our new unit about the immune system. And this unit is set into 3 chapters. The first one is about the organization of the immune system. The second one is about the immune response, how does it take place, and the third one is about the disorders of the immune system. So today we will start talking about the organization of the immune system. But first of all, we have to think of a question. What makes a graft successful and what makes a graft fail? What is the cause that I can take blood from only certain individual and not from all individuals? Actually, this is called the self-marker. It's a self-marker because every one of us has a certain identity that is specific to himself and only himself. And then we can talk about the HLA proteins, which are the first self-markers in document 1, page 114 in your book. Now, what is the HLA? To understand this, we start with the first experiment in document A, which is about grafting. I have in this experiment in front of me 3 types of graft: an autograft, an isograft, and an allograft. An autograft, as you can see, it's a graft in the same individual from one place to another. And an isograft is a graft between 2 identical twins. They are genetically identical. And the 2 grafts, the autograft and the isograft, we can see that they are both accepted 15 days later. And we can see that they have a set of events that take place, which is vascularization. They become richly vascularized, and then they are integrated in the neighboring cells, and then followed by the acceptance of this graft. However, in an allograft, which is a graft between 2 different individuals, genetically different individuals of the same species, we notice that there's a vascularization and they become pink, but then they turn red and the edemas appear. Edemas, which means that we have some dead cells or dead tissues surrounding this graft, and then they become black, dry, and they are rejected. There's also a fourth type of graft, which is not shown here, which is actually called a xenograft. The xenograft is a graft between 2 different species, for example, from a monkey to a human being, or from a pig to a human being, etc. And it is usually also not accepted or rejected without a treatment. Now, what causes the acceptance or the rejection of a graft? It's actually called the HLA proteins. What are the HLA proteins? They are self-identity markers that are found on our cells of the body. These proteins can be divided into 2 classes, HLA class 1 and HLA class 2. HLA class 1, they are found on all nucleated cells of the body. However, HLA class 2 only found on some immune cells like macrophages, and we will see their function in the coming chapter. However, the HLA class 1 and the HLA class 2, they are coded by a set of genes, or by a gene of 6 loci, 1 locus, like a sequence, and 6 loci on the long arm of chromosome 6. This is the long arm of chromosome 6, and I can see that I have 6 loci called DP, DQ, DR, BCA. DP, DQ, DR, you can see that each one is made of 2 letters, so they code for HLA class 2, while BCA, 1 letter, they code for HLA class 1. You might think it's easy then to have 2 individuals who have the same HLA. If they have DP, DQ, DR, BCA, the same sequence, then they would be identical. Actually, this is theoretically true. However, it is so hard to find 2 individuals which are identical. Why? Because these loci, they are formed of a large possibility of alleles that they are highly polymorphic. We say that, for example, in DP we have 72 possible alleles in the population. In DQ we have 49, DR 199, B 188, C 63, and A 82 possible alleles. And you have to remember that of each allele of each gene, I only have 2 alleles, one maternal and one paternal. And by this, I mean that of DP, I might have DPW1 and DPW6, for example, while you might have DPW3, DPW5, which makes us totally different. Even if we share one allele, for example, I have DPW1, DPW6, you have DPW1, DP, let's say 72. We will be still different because the expression of these genes is codominant. And when I say they're codominant, it means that both alleles are expressed at the same time in the same individual, in the same cell. And that's why I'm different even from my parents where I take one allele from one parent and one allele from the other, and that's why it's so hard to find two individuals which are identical in terms of these HLA proteins or in these of MHC genes. The gene is called MHC gene and it codes for an HLA protein. MHC stands for major histocompatibility complex, while HLA stands for human leukocyte antigen. This is the human leukocyte antigen and this is the MHC major histocompatibility complex. So as you see, it's very hard to find two individuals, it's even theoretically impossible to find two individuals which have all of their MHC genes identical, except if I'm talking about identical twins. So how can we make a graft? How can I make a graft which is successful if I cannot find two individuals having the same HLA? Actually, I have to think of an alternative. And in Oxford and in Europe, they tried for different HLA genes compatibility in their kidney transplant between a donor and a recipient. What is this? They started, first of all, we will start with the Oxford transplant, where we are talking about where they considered only the DR allele. They did not consider and they neglected the A, B, C, DP, DQ alleles. They only focused on DR, the compatibility of DR allele between the donor and the recipient. So if the incompatibility is 0, it means that they are fully compatible. It means that if someone has DR1, DR7, the other one has DR1, DR7. They are fully compatible. The success of a graft was 85% regardless of all of the other possible alleles. I'm only considering the DR. And if the DR there's one compatibility, it directly drops and decreases to 70 or 65%. And then if there's full incompatibility, they are incompatible, the success will be around 50 or 60%. As you see, the DR, which is the MHC class 2 gene, is very effective and very very important and essential to be considered as an compatible, and the compatibility should be fully compatible between the donor and the recipient in order to have successful graft. In neurotransplant, they talked about A and B, and then they have different results, which is less important and less significant than that, considering the DR alone. So if there is 0 incompatibility between them, the success is around 72% and then decreases to around 60 to 65%. Nowadays, in order for a graft to be successful, we have to study the compatibility of the donor and the recipient regarding the A and B loci and regarding the DR loci in order to have full compatibility and to have a full success of the graft. I'm talking without a treatment, because we might give immunosuppressors that suppress the immune system and allow the graft to survive for a longer period of time. But without these immunosuppressors, the compatibility should be considered regarding the DR locus and the A and B locus before making a transplant. Okay, so this is basically document 1. It's about the first self-marker, which is the HLA proteins. We can see that these proteins, as I said, they affect the acceptance or the rejection of a graft, where we have 3 types, and only the autograft and the isograft are accepted because they have fully compatible HLA, while the other graft, which is an allograft or the xenograft, they are rejected. I also explained the idea about HLA proteins, which are proteins found on all nucleated cells of the body or on some immune cells. They are referred to as HLA class 1, HLA class 2, but the problem with these alleles is that they are they are polymorphic, and polymorphic means that they have a large number of possibilities in the population, and we only have 2 alleles of each gene. Thank you, and I will be back with document 2 in this chapter.